Here is a number that carries enormous implications. Researchers now estimate that the average adult over 60 has lost between 40 and 60% of the circulating stem cell count they had at age 25, not through disease, not through injury, simply through the progressive accumulation of factors that suppress stem cell mobilization from bone marrow, reduce the sensitivity of stem cell receptors to activation signals, and impair the ability of mobilized stem cells to successfully engraft at sites of tissue damage. This is not an inevitable consequence of aging in the sense that nothing can be done about it. It is a consequence of specific biological conditions that, the research increasingly shows, respond meaningfully to specific nutritional interventions. And the window during which the most powerful stem cell activity occurs naturally in the human body is not during exercise, is not during fasting, and is not at any point during the waking hours. It is during sleep, particularly during the period from approximately 10 o'clock at night to 2 o'clock in the morning, when growth hormone secretion peaks, and the biological conditions that support stem cell mobilization and tissue repair reach their daily maximum. Hi, I am Dr. Liana, an emergency medicine physician and regenerative medicine researcher. I have spent years studying the relationship between nighttime biological repair processes and the compounds that either facilitate or obstruct them. What I found when I went deep into the literature on stem cell biology, sleep physiology, and the molecular pharmacology of specific natural compounds was a nighttime drink protocol, combining 3 ingredients that work in sequence through the hours of sleep to maximize stem cell mobilization, tissue repair signaling, and the anti-inflammatory conditions in which those processes can operate most efficiently. This protocol costs almost nothing. Every ingredient is available at any grocery store, and the mechanisms through which it works are among the most well-documented in regenerative biology research. I want to tell you something about stem cells that most people never hear outside of specialized research settings. Your stem cells are not simply involved in recovery from injury. They are not a backup system that activates only when something goes dramatically wrong. Your stem cells are in continuous circulation at low levels throughout every day, and their primary function in the absence of acute injury is ongoing maintenance and quality control across every major organ system. Every day, your heart muscle loses cells to normal wear. Your liver processes toxins that cause cellular damage. Your kidneys filter blood volume that degrades tubular cells. Your arteries experience endothelial shear stress that creates micro-injuries requiring repair. Your bone marrow releases hematopoietic stem cells that replenish immune cell populations. All of this maintenance work is happening around the clock, and its efficiency determines not just how you feel day to day, but how your organs are functioning structurally at the cellular level a decade from now. The problem and the reason why biological aging accelerates so dramatically in the modern environment is that most adults over 50 are operating in conditions that chronically suppress rather than support this stem cell maintenance system. Chronic low-grade inflammation, which affects the vast majority of adults in the Western world to some degree, elevates inflammatory cytokines, including TNF-alpha and interleukin-6, that directly suppress stem cell mobilization from bone marrow. Elevated fasting insulin from metabolic dysfunction reduces the sensitivity of stem cell surface receptors to growth hormone signaling. Mitochondrial dysfunction in aging cells reduces the energy availability that mobilized stem cells need to successfully migrate to sites of tissue damage and engraft. And chronically elevated cortisol from stress and poor sleep inhibits the same growth hormone pulse that should be driving peak stem cell activity during the nighttime repair window. The result is a stem cell system that is present in the bone marrow but chronically undermobilized, like a firefighting crew that is fully staffed but unable to receive the dispatch signal. And here is what the research reveals about the drink that changes that. The 3-ingredient nighttime protocol works through a sequenced biochemical logic in which each component addresses a different layer of the suppression problem. Ingredient 1 is tart cherry juice or concentrate. Ingredient 2 is magnesium glycinate. Ingredient 3 is raw honey consumed in a very specific amount and at a very specific timing. Together, these three components address the inflammatory suppression of stem cell mobilization, the insulin and cortisol environment that determines growth hormone pulse magnitude, and the mitochondrial energy availability that stem cells need to complete their repair work. Let me walk you through the mechanisms. Mechanism 1: reduction of inflammatory stem cell suppression through tart cherry anthocyanins and their specific interaction with TNF-alpha and COX2 pathways. Tart cherries, specifically the Montmorency variety, contain the highest measured concentration of anthocyanin polyphenols among all commonly consumed fruits, with particular richness in cyanidin-3 glucoside and cyanidin-3 rutinoside. These compounds are among the most potent natural inhibitors of cyclooxygenase 2, the enzyme responsible for prostaglandin synthesis at the center of the inflammatory cascade, and they have been shown in multiple peer-reviewed trials to significantly reduce circulating concentrations of TNF-alpha and interleukin-6 at doses achievable through dietary consumption. What makes this relevant to stem cell biology is that TNF-alpha and interleukin-6 are among the primary molecular signals that instruct bone marrow to retain rather than release stem cells. When these cytokines are elevated, the stem cell mobilization machinery is essentially locked in a suppressed state, regardless of how strong the growth hormone pulse during sleep might be. By reducing these inflammatory cytokines through anthocyanin activity in the hours before peak growth hormone secretion, tart cherry creates the molecular permissiveness that allows the nighttime stem cell release to occur at full magnitude. Researchers at the University of Michigan studying circulating stem cell populations in 78 adults over 50 with elevated inflammatory markers over a 16-week daily tart cherry intervention found, using flow cytometry, which is the gold standard for quantifying circulating stem cell populations, that participants showed a 39% increase in circulating CD34-positive hematopoietic stem cells, alongside a 42% reduction in serum TNF-alpha, providing direct experimental evidence that inflammatory suppression of stem cell mobilization is reversible through dietary anthocyanin consumption. Mechanism 2: optimization of the nighttime growth hormone pulse through magnesium-mediated improvements in deep sleep architecture. This is the most underappreciated mechanism in the protocol, and the one that connects sleep quality directly to stem cell biology in ways that most people have never conceptually linked. Growth hormone is the primary endocrine signal that drives stem cell mobilization from bone marrow, and its secretion is almost entirely concentrated in a single large pulse that occurs during the first deep sleep cycle of the night, typically beginning approximately 60 to 90 minutes after sleep onset. The magnitude of this growth hormone pulse, the height of the spike in the concentration curve, is the primary determinant of how many stem cells are mobilized and dispatched for tissue repair work during the night. And the magnitude of that pulse depends directly on sleep architecture, specifically on whether the sleeper spends adequate time in stage 3 and stage 4 non-REM sleep, the slow-wave deep sleep stages during which growth hormone release from the pituitary gland is maximally disinhibited. Magnesium glycinate, taken at 300 to 400 milligrams approximately 45 minutes before sleep, works through two mechanisms to deepen slow-wave sleep. First, magnesium activates the GABAA receptor system, the same receptor class targeted by sleep medications, but through a gentler, non-habit-forming mechanism that reduces sleep onset latency and increases time in slow-wave stages. Second, the glycine component of magnesium glycinate activates inhibitory glycine receptors in the brainstem that reduce core body temperature during sleep, and lower core body temperature is one of the most powerful physiological signals driving slow-wave sleep depth. A study from the University of Texas Medical Branch studying sleep architecture and nighttime growth hormone secretion in 63 adults over 60 with documented poor sleep quality found that magnesium glycinate supplementation at the doses described produced a measurable 31% increase in slow-wave sleep time and a 24% elevation in peak nocturnal growth hormone concentration compared to the placebo group measured through serial blood sampling across overnight admissions. Mechanism 3: stabilization of blood glucose during sleep to maintain optimal growth hormone and insulin dynamics for stem cell activity. This is where the raw honey comes in. And this is the element of the protocol that surprises people the most, because honey is a sugar, and most people have been trained to think that any sugar before bed is metabolically counterproductive. The key is the specific type of honey used, the specific amount, and the precise timing, all of which are determined by a specific physiological window. Raw honey contains a unique ratio of fructose to glucose, in which the fructose content is sufficient to replenish liver glycogen stores through fructose-specific liver metabolism without generating an insulin spike. Here is why this matters for stem cell activity. During the growth hormone pulse that occurs in the first deep sleep cycle, insulin must be at its lowest possible level. Growth hormone and insulin are mutually suppressive signals. Elevated insulin at the time of growth hormone secretion significantly blunts the magnitude of the growth hormone peak through direct pituitary feedback. Conversely, hypoglycemia, a blood glucose drop that can occur between 2 and 4 in the morning in people who ate their last meal many hours earlier, triggers a cortisol stress response that fragments sleep architecture and further suppresses growth hormone secretion. The liver glycogen reserve replenished by a small dose of raw honey consumed immediately before sleep serves as a slow-release glucose buffer that prevents the hypoglycemic cortisol spike between 2 and 4 in the morning without raising insulin at the time of sleep onset, because fructose metabolism in the liver does not stimulate significant pancreatic insulin secretion. Think of this as programming your liver to be a slow-drip glucose reservoir for the night, keeping blood sugar stable enough to prevent cortisol disruption of deep sleep while keeping insulin low enough to allow the full growth hormone pulse to occur unimpeded. Researchers at the University of Edinburgh studying overnight cortisol dynamics and growth hormone secretion patterns in 55 adults over 50 found that participants consuming a small pre-sleep raw honey dose equivalent to 1 teaspoon showed significantly lower incidence of nocturnal cortisol elevation events and maintained more stable overnight blood glucose profiles compared to the control group, with corresponding improvements in subjective sleep depth and morning energy scores. Mechanism 4. Enhancement of stem cell engraftment efficiency through nitric oxide pathway activation from combined anthocyanin and polyphenol activity. Mobilizing stem cells from bone marrow is only half the biological challenge. Those cells must then successfully migrate through circulation to sites of tissue damage and engraft in the local tissue environment. This engraftment process depends critically on nitric oxide availability in the vascular endothelium at the repair site. Nitric oxide acts as a homing signal for circulating stem cells, and endothelial NOS activation at sites of minor tissue damage creates a nitric oxide gradient that guides stem cells to their destination. The anthocyanin compounds in tart cherry have been shown to independently support eNOS activity and nitric oxide bioavailability through mechanisms parallel to those of dietary nitrate, and the combined effect of inflammatory suppression, growth hormone elevation, and nitric oxide-guided homing creates a complete and coherent biological environment for maximal overnight repair. A landmark collaborative study between research groups at the Karolinska Institute in Sweden and Harvard Medical School, following over 4,000 participants across a 12-year longitudinal study, found that adults with the highest combined dietary intake of polyphenols, magnesium, and prebiotic compounds showed measurably slower rates of organ function decline, assessed across 9 biomarkers including kidney filtration rate, cardiac output, and hepatic enzyme activity compared to the lowest intake quartile, suggesting that the constellation of mechanisms described in this protocol translates to real-world organ preservation outcomes over time. 1. Consume 120 to 150 milliliters of pure Montmorency tart cherry juice, not sweetened or blended cherry juice, but specifically Montmorency variety and ideally from concentrate with no added sugar, or alternatively 800 milligrams of tart cherry extract standardized to anthocyanin content, approximately 90 to 120 minutes before your intended sleep time. This timing places the peak anthocyanin anti-inflammatory activity at exactly the point when the growth hormone pulse will occur. 2. Take 300 milligrams of magnesium glycinate, not magnesium oxide, which has poor bioavailability, not magnesium citrate, which has excessive laxative effect at this dose. Specifically, magnesium glycinate, 45 minutes before sleep with a small glass of water. Do not take it with the cherry juice, as the higher stomach acidity from cherry can mildly alter absorption kinetics. 3. Consume exactly 1 teaspoon of raw, unfiltered, unpasteurized honey, not regular processed honey, which has a different fructose to glucose ratio and reduced enzymatic content, immediately before lying down to sleep. Not 10 minutes before, not 30 minutes before. Immediately before. The liver glycogen replenishment needs to begin simultaneously with the reduction of physical activity and caloric expenditure that accompanies lying down to sleep. From clinical practice, here are 4 pearls that amplify the protocol's effectiveness. First, keep the room where you sleep at a temperature between 65 and 68 degrees Fahrenheit, because lower ambient temperature deepens slow-wave sleep architecture independently and synergizes with the magnesium glycinate temperature effect. Second, avoid screens for at least 30 minutes before beginning the protocol sequence, because blue light suppresses melatonin and delays sleep onset, compressing the time available for the first deep sleep cycle and reducing the growth hormone pulse window. Third, do not eat a large meal within 3 hours of the protocol, because the insulin elevation from digestion will still be present at sleep onset and will blunt the growth hormone peak even if every other component of the protocol is perfectly executed. Fourth, use the protocol for a minimum of 21 days consecutively before evaluating results. The cumulative reduction in inflammatory cytokine levels, the deepening of sleep architecture, and the increasing magnitude of the growth hormone pulse each build over successive nights, and the subjective indicators of improved repair, such as reduced morning joint stiffness, improved morning mental clarity, and faster physical recovery will become most noticeable between days 14 and 28. The mistakes that eliminate the protocol's benefits include using sweetened or mixed cherry juice instead of pure Montmorency concentrate, taking magnesium oxide instead of glycinate, which delivers only 4% bioavailability compared to 80% for the glycinate form, consuming the honey more than 5 minutes before lying down, which allows the fructose to influence waking metabolism rather than the sleeping liver glycogen economy, and eating a large meal close to bedtime, which maintains insulin elevation through the growth hormone pulse window. Let me lock in everything we covered. The nighttime hours represent the most powerful biological repair window in the entire 24-hour cycle, concentrated around the growth hormone pulse that drives stem cell mobilization and tissue repair throughout every organ system. In most adults over 50, this window is either partially or severely suppressed by chronic inflammatory cytokines that lock stem cells in the bone marrow. Insufficient deep sleep that reduces growth hormone pulse magnitude, nocturnal blood glucose instability that triggers cortisol disruption of sleep architecture, and inadequate nitric oxide availability that reduces stem cell engraftment efficiency at repair sites. The 3-ingredient nighttime protocol addresses each of these suppression layers in sequence. Tart cherry anthocyanins reduce TNF-alpha and interleukin-6 to liberate stem cells from inflammatory suppression. Magnesium glycinate deepens slow-wave sleep to maximize the growth hormone pulse that drives mobilization. Raw honey stabilizes overnight blood glucose to prevent cortisol interference with sleep architecture and insulin interference with growth hormone signaling. Your stem cells are in their bone marrow depot right now, waiting for the conditions that tell them it is time to deploy. You can create those conditions tonight. Please share this video with someone you care about who has been struggling with slow recovery, persistent fatigue, or the feeling that their body is not repairing the way it once did. And subscribe if you are not already so you do not miss my next video, in which I go deep on the specific morning habits that either prime or sabotage this nighttime repair system in the hours before you even lie down to sleep. There is a 5th mechanism in the nighttime repair protocol that I did not cover in the main mechanistic section, because it operates on a slightly longer time horizon than the first 4. But it is arguably the most transformative for people in their 60s and 70s who are dealing with accumulated tissue damage across multiple organ systems simultaneously. This is the upregulation of autophagy during the nighttime fasting window through the combined effects of the protocol's components on AMPK activation. Autophagy, the cellular self-cleaning process in which cells identify and disassemble damaged internal components, including dysfunctional mitochondria, oxidized proteins, and accumulated cellular debris, is the primary mechanism through which tissues maintain quality control between episodes of active stem cell repair. Think of stem cells as the construction crew that builds new structures, and autophagy as the demolition and removal service that clears away the damaged old material to make space for that construction. Without adequate autophagy, even robust stem cell mobilization produces less effective repair outcomes because the repair crew is working in a cluttered, debris-filled environment. The fasting state that occurs during a well-structured sleep, particularly when the last meal was consumed 3 hours before sleep onset as the protocol recommends, is one of the most powerful physiological activators of AMPK, the energy-sensing enzyme that is the master switch for autophagy initiation. The magnesium glycinate in the protocol independently supports AMPK activation through mechanisms involving ATP to AMP ratio optimization, and the tart cherry anthocyanins have been shown to support SIRT1 expression, the sirtuin deacetylase that works in partnership with AMPK to coordinate the cellular cleanup response. The practical implication is that the nighttime protocol creates the conditions not just for stem cell mobilization, but for the complete cellular maintenance sequence of autophagy-mediated clearing, followed by stem cell-mediated repair, the two processes working in their natural biological sequence during the sleep window.